Thank you. Good afternoon, everybody. Very happy to present in this kickoff meeting of NEMI platform seminars. Since I didn't actually know who the audience are, I kept it free for myself to go deep in some slides and actually jump over some topics. I see the first problem that is probably from my side, that my slides are running themselves without me changing them. As a preface, let's say, I would like to start that in my research, I am basically focused on studying material formation, which means that if we consider the material formation in different scales from primary particles going through yes, I'm looking for pointer options, exactly, going through particles and then having the particles organized into structures based on their own interactions, which can be probably coming from the shape and the attractions of the particles, or coming from the confinement that causes or manipulates the actually the assembly of the particles into bigger structures. So what I do is that, as you can see from this beautiful theoretical study, there are many possibilities for the particles based on their shapes, morphology, basically, and with the tuned interaction, you can put them into several different crystalline or pseudocrystalline structures. I don't know why this happens, but it is automatically changing the slides. Okay, I go back. Then I would like to, before going to the details of how I look and how I aim for these different structures, I would like to mention for the people who were not familiar with the colloidal assembly field, that it is actually an important field in research. If you look in the scopus of at the number of publications that are coming out over the years, you can see with the term colloidal assembly and colloidal self-assembly, every year we have something like 5,000 papers just talking about colloids. And if we get more specific to colloidal self-assembly, we have like 300 papers coming out. So meaning that there are many people trying to find out how to make these structures, how to get benefits from them, and how to actually improve them, the ones that they are. So it is a very important field to work. And for the start, I would like to mention a specific paper from Dong from 2010, where they first got binary system, a binary crystal at the interface of the solvent and air due to the assembly and the drying of these particles, where you can see they could beautifully put two different particles together and get these assemblies. But enjoying this beautiful picture, we should know that the number of layers of the structured materials they could see here is actually 1, 2, but the solvent is not non-aqueous, and they could only see the analysis of the final coating or structure. But they don't know what has happened in details to get this structure. So if you wanted to move from this example to other material formations, binary material formations, it would be a little bit difficult. So when I started in Eindhoven, I was supposed to make hybrid materials, ordered hybrid materials from organic and inorganic uh, um, precursors, let's say. Uh, and Therefore, I had to do something with the limitations of the previous works in the literature. So the the thing that we decided to do, the way that we decided to approach this project was actually doing time-resolved cryo-TEM and later on, or beside that, developing liquid phase electron microscopy. Again, since I didn't know if we have anybody unfamiliar with electron microscopy, I have put this slide to share that what is time-resolved cryo-TEM. It is actually when you do your multi-scale the material formation study, you do experiments and you do one experiment, and at this a specific time you stop your measurement with plunge freezing, and you put your sample into this beautiful microscope to study the structure of the material at that time point. And if you want to study the process, you have to do that experiment several times and do the plunge freezing every time to get a snapshot or several images of your material at that point. And this is actually a very time-consuming process or analysis, because if you have like a project where you have many parameters and different precursors and many steps in a hierarchical organization, so how many samples do you need to do that? And considering that all of these samples or processes happening at a slightly different time, so basically if you go with a time schedule that you have, each time that you make your sample, you may miss the right time point comparing to the previous sample. So you need to repeat and repeat these cryo-TEM experiments to be able to put the pictures and the snapshots of a process together. Yeah, that cost me 5 years for this project, where we hypothesized and planned to do the assembly of these hybrid materials like this, that we start with two different precursors, silica nanoparticle of 30 nanometer and polymer latexes of 100 nanometer. And then we thought that as I showed in the example, we do control drawing to see whether we can get a three-dimensional structure from that, or we go through an intermediate step, making a more complex building block, which we later called them supracolloids, and then try to concentrate them into these structured materials, hoping that we can do it in water and we can get thicker materials, not just a few layers. And then we hope that with electron microscopy we are able to analyze them. So the system we chose actually needed a switch because if we wanted to put the silica and latex together in attached position to form our supracolloid, we needed this attraction to happen via, for example, pH switch in our case. So we functionalized our silica materials with different ratios, which later on happen to be the key to shape our supracolloids in a perfectly covered material. As you can see here, we change the charge densities following the curves of pH switch and the zeta potential of the system, and then with a specific ratio of factors, the amino functionalities on the surface of the silica, we could reach almost fully covered supracolloid in our system. But we were not sure whether we really have them fully covered or not. Therefore, we did a lot of tomography analysis, and then from that we analyzed the surface structure, surface arrangements of the silica particle. You can see on the right here to see whether they are, first, what is the structure, and second, whether they are fully covered or not, which compared to some theoretical studies of Mansfield before, as you can see in the middle, when you have a curved surface and you want to cover that with some particles, the number of the coatings, coating particles to the number of to the radius of that binary particle, we call them here superparticles, should follow this curve. And you can see the experimental data from us falls are close to that and on the other side. So we are not exactly matching the theories, but we are very close to what it should be. And yeah, there is always a error for the theories compared to the experiments. So we could analyze them. We could see the structure of the silica particles on the surface, and actually this hexagonal or 6 nearest neighbors for the majority of the particles sort of confirms that we are almost fully covered with silica particles. So once we made our building blocks, the next step for us was to actually go for the drawings and finding out whether we can make the 3D structure material or not. The drawing would happen, as you can see on top, by putting a very small droplet on the TM grid and then controlling the humidity in a Vitrobot again take snapshots via tomography by plunge freezing at different steps. And I'm very happy to mention here that there were also some interesting moments in this process of drawing, that you could see the refractive index matching would happen just before a fully dried droplet or coating forms on the TM grid. And then from the bulk material to the middle of the drying process, and almost close to the dried state, we could do tomographies and analysis of the particles, which we actually saw some important phenomena happening. So if you look at this middle cluster in deep, you can see that there are only one layer of silica standing between the two particles, and that brought us to think whether a model for this clustering could be that the two supracolloid come together, and then we get some rearrangements of the silica particle on the surface between the two particles, and therefore we need to where the excess silica goes. And a deeper look could actually show us that the excess silica is there on on a corner of this cluster and the other clusters. So we found that packed silica layer surrounded the cluster with only one layer of silica at the interface in these moments. And then this would actually bring us the hypothesis that, yeah, we don't get ordered material as we saw in the last tomographies, because we get clusters in between, and these clusters introduce some silicas in a space that we don't want that, and that causes the disordered material at the end. And then we thought, if we use all of these knowledge that we've got to sort of arrange the repulsion between the particles, so that when we dry them, the rearrangement of silica or clustering of the particles is not happening. Maybe we are able to get an ordered material at the end with a single layer or a double layer, depending on how much the space in the triple organization of the particles would give us. So we also asked for some help from collaborators, Professor Alex Ruth of Cambridge. He wrote a kinetic model for the assembly, telling us how we should approach this assembly and when does this clustering happening in our systems. According to his calculations, the maximum of the fully covered particles without the clustering in the system would happen at 1800 seconds since the mixing happens. And our other collaborators at Utrecht, under supervision of Marjolein Dijkstra, actually calculated for us how we should arrange the interaction of the particles of the small silica particles and the large latex particles to be able of making these supracolloids, and also with suggestions of where to go to basically keep them separate from each other over drying. So more repulsion of them when we have them in a drying process. So learning from these, we managed actually not to just make a structured materials with the original 130 nanometers, which was not, I would say, structured due to the dispersity of the sizes of the particles that we had. But we could a little bit change the size ratios and then get really ordered materials, as you can see here. So, and then we analyzed them via stem tomography in some cases, and we could see the order inside the material. So you can see several layers of the material, nicely compact, and then having the silica particles in between. And these silica particles are so stable that even after calcination and removal of the polymer, you can still see the structured materials in some cases. So I see that I missed the size ratios in this picture when I picked them up, because I don't have the scale bar mentioned here. The scale bar here is a micrometer, so you can see how big the domains of a structured material could be. But talking about cryogenic into the naming audience, I would like to mention a very special moment in this project where we succeeded to capture the particles very closely. I mean, after adjusting the repulsion interaction, but still not contacting, which or let's say not forming a cluster, uh, which was actually from a sample that later on resulted in this beautifully ordered material. And when we look into the analysis of the particles of this sample, it was very interesting to see that the silica particles sitting on one supracolloid particle are sometimes as close uh, to the silica particles of the other supracolloid. So the material has gone this concentrated, but still no rearrangement of the silica particles happened into the thing. So, which means that we sort of confine the replacement of the particles that happens at the final drying moments in a very narrow region where they cannot actually escape and make phase separation. So this was the sample that helped us understanding where to go and how gave us hope to go for high repulsion forces and then hoping for the ordered material at the moment at that moment in the project.
Mohammed,
Which
Mohammed,
yes,
you're at we're coming up on 22 minutes, so you have another 3 minutes left.
okay. So having, I mean, finding this actually, I would like to, uh, I would like to say that it is cryo electron tomography is very important and very helpful in analyzing the structure, analyzing the process, but finding the right moment to take the imaging is quite difficult. And therefore, I would like to tell myself and others the benefit of going towards liquid phase electron microscopy, which brings high resolution in case of using graphene sandwiches. And with graphene sandwiches, you can actually save your samples compared to cryo samples 7 to 10 times more in front of the exposure. And recently, Pauline van Dorsten have provided and published a new way, which is very easier and more productive in preparing graphene cells. She has also tracked the place of water. Yes. So the analysis of the pockets, which I would like to mention today, shows that you get like pockets of 100 to 200 in this form, nanometer. But if you want to do work with bigger materials, it might be a little bit of a problem. So recently, we have used graphene cells to put from immunoglobulin, collagen formation, and even the Deinococcus bacteria, and the flexibility of the graphene sitting, sandwiching the material and having the form of the material is actually very flexible and helpful, but the dynamics of the material inside could be a sort of a problem. Very recent experiments that I could do despite the corona time show that if we replace graphene, one of the graphene layers with carbon, we can actually make huge water pockets. And if we do surface treatment a little bit, we can make channels of matter, and you can compare these really huge channels with what we had before. We can still follow up processes with electron diffraction analysis, and a very surprising moment or experiment was that it's my last two sessions slides. So could be the, I don't know why it's not showing, but you can see the supracolloids trapped in these water particles. So with that, we are coming to the moment.
We may be able to introduce the movements that we want, and from that we are combining the abilities of the silicon nitride cells, the flow cells, and control pressures, together with the high resolution that we get from graphene.
It seems that this is coming from your side. There's nobody else with the microphone open.
Yeah, that's true. So that was a sort of recording on my slide. I don't know where that comes from, but yeah, considering all of that, it said what I wanted to say. We at the moment we are trying to make a spacer that we can actually form the graphene cells in a way that we can move the particles inside using the bubbling. So with that, the summary of what I wanted to say today is that the extensive cryo-electron tomography of colloidal acid resulted in modeling the clustering process of the supracolloids. Controlling the repulsion of supracolloids resulted in 3D hybrid nanostructures. Considering the benefit of GLCs, replacing graphene with thin carbon is useful for some material dynamics studies, and a design framework for GLC, which is the next thing to do, can improve the use of nanofluidic GLCs to monitor reactions and processes via liquid phase electron microscopy. And with that, I would like to thank the people who helped me over the years in this study, and I would like to thank you and your audience for your attention.